Hey everybody, welcome back to General Biochemistry Lecture. We are starting Chapter 6, which is on enzymes. This is a very large chapter, so I'm splitting it up into two videos. We're not going to go super heavy into the math and deriving equations, but I'll point out what you need to know and kind of how you need to be thinking about this information on enzymes. It's one of those things that you could spend a lifetime studying. We don't really have a lifetime. We have like a week. So we're going to simplify it a little bit, but I want you to have a great appreciation for enzyme kinetics after this chapter. These are the key principles. Enzymes are super powerful biological catalysts, and you're going to see that by the end of this lecture. You can read through the rest of these principles and be able to expand on these. But I think that you'll really understand just why enzymology, which is the field of studying enzymes and their chemistry, why enzymology is such a powerful, powerful subject of study. So first we're going to introduce enzymes. What are they? What do they do? So back in 1897, it was demonstrated that there are these cell-free yeast extracts, which pretty much means you don't have intact cells. You have the cell innards, okay, the cell goop, everything that's in the cell without the actual membrane around it. And you could take that extract and ferment sugar into alcohol. And that fermentation was promoted by all the things that were in the cell, even if you didn't have the cell intact. So the name enzymes were given to those molecules that were detected. Most of the enzymes that you're going to encounter are proteins. There are exceptions, but most of them are proteins. And because they're proteins, they have a native protein conformation. Without that conformation, they are not catalytically active. So the structure of the protein is integral to its function as an enzyme. The characteristics of enzymes is all over the place. They vary quite drastically. Molecular weight can be small to really huge. Some enzymes, most enzymes really, require additional chemical components like a cofactor or a coenzyme. We're going to give some examples of both of these, but a cofactor, we're talking about inorganic ions, metals. Coenzymes, we're talking about more complex organic or metalloorganic molecules that help to carry specific functional groups. So when you think about, this isn't exactly an enzyme, but we talked about hemoglobin for chapter 5, and it has something that is definitely not an amino acid that's bound to it. That is the heme group. And it's got the iron and the protoporphyrin. So that's something that you could think about. There are all kinds of molecules that are bound to enzymes and just proteins in general that help them do their job. These are some examples of inorganic ions as cofactors. You don't need to memorize this, just understand the concept that you can have various ions acting as cofactors. And the same for the coenzymes. No need to memorize this, but this just gives you an idea of all the different types of coenzymes that you might see. When we get to metabolism and we're talking about the mechanisms of each of these pathways, then we'll really dig into the enzymes and their pathways, the coenzymes or the cofactors that they use. But for now, we're just kind of giving you the overview of these things exist. Here are some examples. There are some additional chemical components that enzymes can have. There's the prosthetic group, and that just means that you have a coenzyme or a metal ion that is super tightly bound, or even covalently bound in some cases, to the enzyme. The holoenzyme is the whole enzyme. So it's catalytically active, and it's got all the things it needs. The protein component, the coenzyme, whatever, metal ions, all of those things together create the holoenzyme. The apoenzyme or the apoprotein, That just means the protein part. So it's the protein minus the cofactors. You can imagine that the apoprotein probably isn't going to be active if it requires some metal or some other cofactor bound. These are the classifications that are recognized. We're not going to go too heavy on this in this chapter, but we will revisit this table to look at the classifications of enzymes that are in the metabolic pathways that we're going to be studying coming up in the next few chapters. So put a pin in this. I'd recommend having this table in your notebook, or if you have the book, to kind of tag it so you can go back to it over and over again. But enzymes are divided into 7 classes, and each of those classes has subclasses that's based on the different types of reactions that they catalyze. Each enzyme is assigned an enzyme commission number, the EC number, and it's a systemic name. So that way you can look up any enzyme and you'll find that information, that name, that number, and every time you see that number, you're talking about that enzyme. In addition to that number, there's also a trivial name. So what you would call it, it's kind of like the name that everybody knows you by. Okay? So think about the EC number as being the name that's on your birth certificate, and then the trivial name is the one that everyone, calls you. Maybe it's your nickname, maybe people call you by your middle name, whatever it is. How do enzymes work? The answer is, well, no. Enzymes have a little place called an active site. And that active site provides the exact environment that will allow a particular reaction to occur very rapidly. So here we've got a protein, it's an enzyme, and that red patch represents the active site. And that's just all the amino acids, and if there's any cofactors or coenzymes, all of that is in that tiny little active site. And there's also a substrate bound. So the substrate binds to the active site. That's the place where all the chemistry happens. And notice how small that active site is in reference to all the entire rest of the enzyme. So that whole enzyme is there just so that it can protect that active site and provide the correct environment. The substrate is the molecule that's bound to that active site, and that's the thing that's going to have some work done to it, okay? It's going to have some chemistry happen, and it's going to leave changed into some product. If we're looking at a simple enzymatic reaction, the first step, we've got the enzyme, which is E, binding to the substrate, S. And that is a reversible reaction. So that's what those double arrows are, reversible. We form an ES complex. After that, you have chemistry happen, and you form the product. That oftentimes is also reversible. Enzymes can do the forward and reverse reaction, but it's a matter of which one is more favorable, and there's all these other things to consider when we're talking about whether or not the formation of product is reversible or not. Okay. But in this example, we're calling it reversible. So then you form this enzyme product complex. And from there, the product has to leave the enzyme, wave goodbye, and they're separate. So you have enzyme plus the product. So the two transient complexes that we form are ES and EP. This is just a simple enzymatic reaction. Any enzyme that can catalyze the reaction of the substrate going to the product can also catalyze the reverse, as I said. Enzymes accelerate the conversion of substrate to product. They're catalysts, which means that they're not used up in the process. So that's what a catalyst is. Something that accelerates the process, but is not, a component that gets used up. And most importantly, the equilibrium point is unaffected. Catalysts do not affect the equilibrium. They affect the reaction rates. Probably going to say that several times throughout this, but it's a very key thing to understanding enzymes. Enzymes do not affect reaction equilibria. They just affect the rates. And the way that enzymes enhance rates is pretty crazy. We're talking about orders upon orders of magnitude that reactions are enhanced. So if you have a reaction that takes place on the benchtop and it's really hard, it takes a long time, an enzyme may be able to do it 10,000 times faster. Which that number, 10,000 times faster, that doesn't even make sense. But what we're talking about is something that takes, maybe a second, if that, versus something that takes 10,000 seconds. Just think about that for a minute. I don't even know what 10,000 seconds is in terms of minutes. I'm not going to do the math. You can if you want to. But the comparison is quite drastic when you think about it in terms of time. Again, you don't need to know what these enhancements are. Just have an appreciation for the fact that enzymes enhance the reaction rates quite a bit. Now we're going to get into how they do this. We're going to introduce some more terminology, and you may remember this from Gen Chem perhaps, when you're talking about ground state and transition state, perhaps even in organic when you're talking about different mechanisms. So we're going to start with talking about ground state and transition state. So the ground state is where you're starting for the forward or reverse reaction, but it's where you're starting from. So here's our ground state. The substrate has some kind of energy associated with it. That's the ground state. Then we have the transition state, which is all the way uphill. The transition state is the point where the substrate or the product are equally likely to form. So you've done some kind of chemistry, you've got some distortion of bonds, and you can either fall downhill and make the product, or fall downhill the opposite way and go back to the substrate. When we're talking about these transition states and the energies associated with them, we're talking about delta G, the free energy. And the energy of the transition state is usually pretty high. So we have to go from this substrate to some high energy transition complex where bonds are being broken and formed. And then we have to roll downhill where we've made a product with new bonds formed, and that product gets released. So that transition state has a lot of energy. It has a high energy requirement. Since we're talking about biochemistry, we're going to use the biochemical standard free energy change. So the only difference is that there's a little prime here, and I'm going to highlight that. And what that prime means is that we're looking at the free energy change at pH 7. pH 7 is biologically relevant. The activation energy, that's the difference between the ground state energy level and the transition state energy level. So what I just highlighted, that is the activation energy to go from the substrate to the product. What enzymes do in all catalysts is that they lower the activation energy and increase the reaction rate. So if our traditional reaction on the benchtop with no enzymes, no catalyst whatsoever, if that energy is really big, right? Look at this arrow. It's huge. That's our activation energy of the uncatalyzed reaction. With an enzyme, we can make that significantly smaller. So that's what enzymes do. They lower the activation energy required to go from substrate to product. In between the road from substrate to product, you have reaction intermediates. And we talked about the intermediate, their transient complexes, ES and EP. That's enzyme with substrate bound and enzyme with product bound. Notice that those are in valleys. So the energy associated to make the ES complex, ES is stable, EP is stable. The transition state is not. The rate limiting step is the step in any reaction that has the highest activation energy. That is the step that determines the overall rate of the reaction. So that reaction is only as fast as its slowest step. That's definitely something you'll want to understand and wrap your mind around, what the rate limiting step means, and that it determines the overall rate of the reaction. Rate limiting steps are great for regulating metabolic processes, and oftentimes you'll see multiple rate limiting steps within a process. So we'll point those out as we get to metabolism, which ones are the rate limiting steps. So you'll need to have a good understanding of that concept before we move on. The activation energies that we're talking about, those are the barriers to chemical reactions. So looking at a reaction on the benchtop versus with a catalyst, the activation energy, that barrier, is much lower with an enzyme or other catalyst present. Enzymes have developed lower activation energy selectively to increase the rates for all the reactions that you need to survive. So enzymes are not going to, develop or evolve to do a reaction that doesn't actually benefit sustaining life. So all the reactions that are occurring in your cells right now as you're listening to this lecture, they're performed by enzymes that have had a lot of time to figure out the best way to do the work. Now we're going to get into some math. We've been talking about rates and reaction rates. Now we're going to give some more terminology there. So we're going to talk about rate constants and rate equations. The rate of any reaction is determined by the concentration of the reactants and the rate constant, which we always go with a K and it's italicized. For the unimolar reaction, which means that it's just dependent on the one substrate, and that converts to product, the rate equation is, you have the velocity V is equal to the rate constant K times the concentration of the substrate. So that is just a simple rate equation. It's also a first order reaction. So that means that it only depends on the concentration of one thing, S. The units of K will change depending on the order of the reaction. For a first order reaction, it has the units of reciprocal time, like seconds to the -1 is usually a pretty common one. A second order reaction depends on the concentrations of two different compounds, or it could be two molecules of the same compound, like you need two S's to make the product P. And the units that it has is per molar per second. So your rate equation becomes V, the velocity, is equal to K, which is that rate constant, times the concentration of each substrate. So S1 and S2. You don't need to memorize these rate equations. If that's how it works for you to understand what happens, then sure. But it helps to be able to take a reaction like this and write the rate equation. So you know that it's dependent on the concentrations of S1 and S2, and that there's some kind of rate constant associated with that. And you should be able to write that equation. But why is it that enzymes are able to lower the activation energy? The key is the binding energy that's derived from the enzyme-substrate interaction. All those non-covalent interactions that we were talking about in Chapter 2. Hydrogen bonding, ionic interactions, hydrophobic effect. All of those things create binding energy that helps to lower the activation energy. It offsets the activation energy required to do the chemistry. Very important concept here. You know, we spent like 3 seconds on this slide, but this concept is really important to understanding how enzymes work. Now, there was the lock and key hypothesis, where enzymes are structurally complementary to their substrates, but it turns out that that's wrong. That would actually make a really terrible enzyme if it were complementary to the substrate. And we'll talk about why. In your book, there's the example of having a metal stick that you need to bend and break into two, broken sticks. And that's the product. Okay. Without any enzyme, you have this really big barrier, really high activation energy. Now let's propose that we have an enzyme that's complementary to the substrate. It's going to bind that metal stick really, really well, but it's not going to want to break it. It's not going to want to make any new interactions because it's already fully complementary to the substrate. This is a very stable complex. So there's no energetic drive to actually break this interaction. There's no need. The enzyme and the substrate are happy. They don't want to break apart. They don't want to make anything new. They just want to sit there and hang out together. But if you have an enzyme that's complementary to the transition state, then we can work. Why? Because there's some weak interactions here. And that helps you to form ES, the enzyme substrate complex. But there's more to be had here because there are more interactions as that enzyme and the substrate kind of have that induced fit thing going on, where the substrate kind of bends to optimize those interactions. So that's what's happening here. We optimize the ES interactions. And what that looks like is the transition state. You've got bonds in the process of breaking, bonds in the process of forming. And then finally, you roll downhill to make product. Make sure that you understand why an enzyme being complementary to its substrate is a bad idea. You'll probably see a question like that on the exam. Just going to throw that out there. So to recap what we just talked about over the last few slides, if you take the sum of the activation energy that's unfavorable, so uncatalyzed, right, and you add to it that binding energy of the substrate binding to the enzyme and all of those interactions, that gives you a lower net activation energy. So your delta G catalyzed. All the weak binding interactions between the substrate and the enzyme drive enzyme catalysis. So if you need one, take-home slide that covers the last few slides, this one's probably it. And we mentioned that enzymes have an active site, a little spot that has the exact right environment for the chemistry to take place and for the substrate to bind. The binding energy is optimized in the transition state in the active site. And remember, all of these molecules, the substrate, it's got this hydration layer around it. It's got water all around it to keep it in solution. So you have to overcome the interactions between water and the substrate and make it more favorable for the enzyme to bind the substrate. So you have to replace all those interactions and make it so that the substrate is like, oh yeah, I'll let go of the water so I can bind here. This looks like the place I want to be. The specificity of an enzyme is the ability to be able to discriminate between a substrate and a competing molecule, and that is linked to the binding energy. A competing molecule may not have all the complementary things that allows it to bind to the enzyme well, and it may not be complementary to that transition state, so it might go and kind of test the waters, dip a toe in the water, say, oh, it's too cold for me, and then that competing molecule will leave. But a substrate, which enzymes can have multiple substrates, the substrate is going to go dip a toe in and say, oh yeah, this is great. I'm going to get comfortable here. Start stretching out, and then you form that transition state, and then the chemistry happens. So the specificity comes from that binding energy. We talked about the barrier to reaction, right? But there's a lot of different things that contribute to why there's such a high barrier to making this chemistry happen. There's the entropy of the molecules in solution. So when you have the enzyme and the substrate coming together and staying in one orientation, that decreases the entropy of the molecules, and that's not favorable. Then we've got the solvation shell of the hydrogen bonding, with the water and all the different things that are in solution that are potential substrates. Removing that water so that the substrate can bind is also unfavorable. The substrates usually have to be distorted in order for some kind of chemistry to occur. That is also unfavorable. You see the trend here. There's a lot of things that enzymes do that are quite unfavorable. And the last one is that you have to have proper alignment of all the functional groups in that active site for that enzyme to work. So we're talking about, if you have some kind of a general acid base catalysis going on, you need that general acid, whichever amino acid that is, to be positioned in just the right way so that it can donate a hydrogen to the substrate. So having all of those things coordinated and stay in place, unfavorable. Let's talk about these one by one. When you have to restrict the movement of molecules, you're going to reduce the entropy. That's never favorable. But the binding energy constrains the substrates in that proper orientation because of all the interactions that are formed that are favorable. We keep coming back to the same idea of the binding energy being what the driver for enzyme catalysis. So if you're like, I feel like we've said this a bunch of times, you're right, because it's the reason for all the things. When in doubt, the answer is either 42 or binding energy. Maybe not in real life, but it's worth a shot. So what we have in this figure to the right is a series of different reactions. And we go from having two separate substrates that we have to bring together and form one product. Let's say that the rate enhancement for that is 1. Let's say that we've got a substrate that all the functional groups that we need are in that same one molecule. We just have to do a little bit of rotating and twisting to bring them together and make the chemistry happen. Well, that's easier to do because we've only got one molecule. We just have to distort it a little bit. So we can enhance that rate even more. But let's say that we've got a substrate where everything is already in position. We've got that oxygen and the carbonyl already posed to attack the other carbonyl carbon. We just have to hold on to it. That's a lot easier, where everything is already in place and the electrons just have to jump a little bit. So orientation is everything. And that's why enzymes are so great at being catalysts, because they properly orient the substrate with all of the things needed to do the chemistry. So the next thing that enzymes have to overcome, the next barrier, is desolvation of the substrate. And that is just replacing all the water that's structured around the substrate that's, hydrogen bonding with it. And instead replacing those hydrogen bonds of the water with the weak bonds with the enzyme and substrate. So the enzyme has to replace the majority of the hydrogen bonding that the substrate was enjoying with water. It's got to be at least the same. If you're moving from one apartment to the next, you're not going to go from an apartment that has, 2 bedrooms, 2 bathrooms, you've got washer and dryer in your apartment, and then you're going to move into a studio, no washer, dryer. That just makes no sense. Well, the same thing for the substrate. It's saying, look, if I've already got the 2 bedroom, 2 bath, I at least need to get an equally sized apartment. I'd prefer to move up, maybe get a house. And so that's why the enzyme has to overcome this, because the substrate is already happy hanging out in the water. So it needs to provide some kind of interactions that will appease the substrate, aka binding. We said that the substrate has to undergo some kind of distortion, right? Because when you're doing chemistry, you're breaking bonds and forming new ones. Those electrons have to be redistributed. Oftentimes, that requires an unfavorable free energy change. So the binding energy of the substrate to the enzyme compensates thermodynamically for this, which we've already said probably 80 times at this point. We brought up induced fit a little bit, but now we're going to talk about it again. If you have an enzyme and it binds the substrate, there's usually a conformational change to the enzyme itself when the substrate binds. And those weak interactions that the substrate initially forms help induce further changes in the enzyme. And that enhances the catalytic properties of the enzyme. So you have enzyme binding. There's a few weak interactions. And then the enzyme says, oh, I've got my substrate here. Let me whip out some more. Okay, I'm going to twist a little bit. I'm going to let this functional group poke out a little bit more. and I'm going to form some more hydrogen bonding, or maybe do some pi-pi stacking, like interactions, van der Waals, but only because the substrate is present. So that's what we mean by induced fit. Remember that there are all types of catalytic functional groups that aid in the cleavage and formation of bonds by a whole host of mechanisms. And those functional groups can be the functional groups that are the R groups on an amino acid, or it could be a coenzyme, it could be metal, lots of different options here. We're going to talk very quickly about the general acid-base catalysis. This is a very, 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 very common method of catalysis. You've got protons transferred between the enzyme and the substrate, or an intermediate. Without a catalyst, the intermediate would be really, really unstable, because you've got this positive charge here transiently, and you've also got this negative charge here. So this is the transition state. And it is highly unstable. Trying to carry two charges in one molecule? Are you crazy? So this intermediate is going to rapidly break down, form the products again, and say, nope, can't do it. However, if you have an enzyme, you can stabilize that transition state. So that's a big plus. The other thing that an enzyme can offer that just doing it on the benchtop can't really give you is that you're only limited to the hydronium or the hydroxide ions that are present in water. All the things that you can kind of bring to the game. Whereas with general acid-base catalysis, we're talking about R groups on amino acids. So you can think about what amino acids can form, can be proton donors and which ones are acceptors. So which can be an acid, which can be a base in acid-base catalysis. And we can use these weak acids and weak bases other than water to do all the chemistry. So that is a big plus. And between those two things, we stabilize that transition state, and we have other weak acids and bases that we can use to transfer protons. You're going to greatly accelerate the rate of this reaction. I'm not requiring you to know the acid-base catalysis mechanism yet, but as we come across these mechanisms in metabolism, then you will need to recognize these and understand. But for now, we're just talking about the broad strokes. So like I said, a lot of enzymes use acid-base catalysis. And this table gives you a great idea of how amino acid residues can act as general acids and general bases. So I definitely recommend sitting down with this table and making sure that you understand how each of these residues in this box can act as a proton donor and a proton acceptor. Now we have covalent catalysis, and that's where transient covalent bonds form between the enzyme and the substrate. This catalysis is only going to happen when the new pathway has a lower activation energy than the uncatalyzed pathway. So let's say that we want to hydrolyze a bond. That means use water to break a bond, and we're trying to take this compound that has A bonded to B, use water to cut it, and we liberate A from B. That's going to be a really hard reaction to do without an enzyme. But if you have a covalent catalyst, And this X you can think of as your enzyme. That enzyme can replace B in the reaction. So you're going to have this these lone pair electrons attack the bond between A and B, and X is going to replace B in the reaction. So your enzyme replaces B. Now you have this covalent bond between your substrate and your enzyme. And you've got free B. Then you can use water that's coordinated within the active site to liberate A from the enzyme. Liberate, cleave, cut, those are all terms that you're going to see and hear when it comes to catalysis. So we're going to cleave A and regenerate the enzyme. Remember, the enzyme is a catalyst, which means it does not get consumed. So if we stopped at the point of A being covalently bonded to X, now we can't do any more chemistry. That enzyme is not a catalyst anymore. It was a substrate. We have to have the hydrolysis or whatever other reaction necessary to liberate A from X and regenerate that enzyme. Finally, metal ion catalysis. One of my favorites. So if you ever wondered why you need metals in your diet, we're not talking about heavy metals, okay? No mercury. But we're talking about things like iron, calcium, magnesium. These metal ions are key for a lot of enzymes to do their chemistry. So we have a steady diet of these ions because enzymes, just like all the other proteins, have a certain amount of time before they get degraded. So they do their chemistry, they get degraded, and sometimes you can recycle the metal ions that are bound, but you need to kind of have a constant influx to accommodate the fact that you're destabilizing some of these proteins, you're chewing them up, it's called proteolysis, right? So you have to eat your nutrients, your vitamins, all these metals, all the parts of food, because you need all of those parts to actually drive catalysis. So, metals can do a lot of different things. They can help orient substrates by providing, interactions. Stabilize charge. So think about it. You always have ions, iron 2 plus, iron 3 plus, calcium 2 plus. All of those are charged ions, and they can help stabilize a charged intermediate. They can also mediate oxidation reduction reactions because oftentimes you'll see transition metals like iron in an active site, and they can go from iron 2 plus to iron 3 plus and vice versa. Changing the metal ion's oxidation state can help you do redox reactions. So almost a third of known enzymes require at least one metal for catalytic activity, which is mind-blowing to me. There's a lot of enzymes out there. Enzyme activity, which we've been talking about, is dependent on pH. So we briefly talked about that at the end of chapter 2, where we said the environment matters. And so pepsin is a protease that's found in the stomach. and its kind of optimal activity is around pH 2. That makes sense. The stomach, lots of acid. So this enzyme needs to be active in an acidic environment. But what if you took pepsin and put it closer to 6? The activity here is going to be much, much lower. And the pH of the, at the optimal activity gives you an idea of the types of amino acids you might see involved in catalysis. So if you need a low pH, then maybe that means that you need a lot of hydrogens. And maybe you need a hydrogen to be, present on one of your R groups so that it can be donated to something else. Another example is glucose 6-phosphatase. Its optimal activity is somewhere around 7. That makes sense too. It's inside the cell. It's not in the stomach like that. We're not talking about being active in stomach acid. This is just a general phosphatase, which means that it cuts off phosphate groups. So it's going to be active more around pH of 7, like most of the enzymes in your body. Now we're going to get into some enzyme kinetics, and we're going to break up this part into 2 sections. So this is the last part that we're going to cover for this lecture. Enzyme kinetics is just studying the rates of reactions and how those rates are affected by changing experimental parameters. So you can change pH, you can change substrate concentration, you can change temperature. All of those things you can experimentally control and study the rates of reactions. You can study pre-steady state and steady state. Pre-steady state, we're talking about a small snippet in time when the enzyme substrate complex is building up. Now, normally that ES formation is really, really fast. So it's hard to see and you need specialized equipment to do it. We're going to talk about pre-steady state at the beginning of the second lecture. So part 2 of this is going to pick up with pre-steady state. We're going to focus on steady state, and that's where you've got a constant concentration of your ES complex and your other intermediates. Traditionally, this is where you start. You analyze the reaction rates under steady state conditions. And we're going to talk about all of this terminology and what steady state is. When you're studying enzyme catalysis, we're talking about velocities, right? So how much product are we making over time? You can track that. So experimentally, you can control how long the time is, right? So you can measure this in seconds or minutes, whatever is appropriate to your enzyme catalysis. And you have to have a way of monitoring your product formation. Fluorescence, absorbance, all kinds of different ways that you can monitor product formation. If you do that, and you can vary the amount of substrate that you have in solution. So in each little, we'll say test tube, but oftentimes you use a plate reader, like a 96 well plate or something like that. You're going to have enzyme plus your substrate in a pH optimized buffer. So if you have an enzyme that works best at pH 2, then you're going to want to have your buffer around pH 2. And as you do chemistry, you're not going to appreciably change that pH because it is in a buffered solution. So those are the things that you have in your test tube. You form product, which you have to be able to observe using fluorescence, UV, any host of things. And as you do this, you have different concentrations of substrate. So maybe you have 3 test tubes and you have say 1 millimolar, 5 millimolar, and 10 millimolar. You've got 3 different substrate concentrations, and you're going to monitor product formation at all 3 of these concentrations. If you take the tangent of each curve at time 0, that reflects a steady state. So that gives you the initial rate, the initial velocity of the reaction. And we can treat the concentration of S as a constant because it's at the beginning of the reaction. So we're saying, it hasn't really changed. So we can take those initial rates and we can plot them against substrate concentration. So you've got your initial velocity for all these different substrate concentrations. You plot them and you get this square hyperbola. There's a magical constant called the Km, that's the Michaelis constant, that you see for a substrate when you've reached half of your Vmax. The Vmax is the maximum velocity that you could theoretically achieve with this enzyme and this reaction. Now, chances are you're never going to actually see that in the test tube. There's a lot of different reasons why. We're not going to talk about that. But the Km is a special substrate concentration that we're going to talk about some more. But this is the plot that you generate. So you do all of your different concentrations of substrate and you find the initial velocity for each of those. Then you plot the initial velocity on the y-axis against the substrate concentration on the x. And that's how you get this curve. Now, why do we care about that? Well, it's all connected to Michaelis-Menten, which is these 2 scientists did a lot of work with understanding enzymes and proposing steady state. Okay. This is the general theory that they proposed about how an enzyme works. In the first step, you have the enzyme and substrate coming together, and they form a complex, and this formation is reversible, and it's pretty fast. You'll see our rate constants, K1 and K minus 1. The minus means just the reverse reaction. So if we're forming ES for K1, then K minus 1 means the ES complex falling apart. In the second step, the complex breaks down to make enzyme, free enzyme, and whatever your reaction product is. And that reaction is the slower step. So the formation of ES is fast. Formation of free enzyme and product is slow. And that means that the second step is going to limit the overall reaction rate. It is the rate limiting step. So the overall rate is proportional to the concentration of ES. You're only going to be able to observe Vmax, that maximum velocity, when all of the enzyme present in your reaction is completely bound to substrate. Everything is bound to substrate. There's no free enzyme anywhere. It's you release product, bam, substrate. There is no in between. And you know that you've reached that velocity when you continue to increase the substrate concentration and there's no effect on the rate. Now, in real life, you can increase the concentration of substrate so much that you actually start to see inhibition and you see a decrease. But that's kind of an experimental detail, not theoretical. Just throwing that in there to kind of balance it out. But this is the plateau that you're seeing. And I'll highlight the plateau. So this plateau right here, we keep increasing the substrate concentration, but we're not really seeing a change in Vmax. So that's the saturation effect. The relationship between the substrate concentration and the reaction rate can be expressed with the Michaelis-Menten equation, which is right here. So we've got our initial velocity, V naught, that's 0. We say that naught. So if you remember from physics, if you've taken physics already, the Vmax times the concentration of substrate over the Km, which is the Michaelis constant, plus the concentration of substrate. So this is the Michaelis-Menten equation. You'll need to recognize it. I'm not going to ask you to do any math with it per se. If I gave you all of the variables and you needed to solve for one, you should be able to do basic algebra, but you don't need to memorize it. You won't need to do any derivations or anything like that. You'll see the derivation of the Michaelis-Menten equation in your text if you're really interested, but I am not going to require you to know that. I just want you to recognize all the parts of the equation and be able to do some simple math with it. So this equation is actually the rate equation for a one substrate enzyme catalyzed reaction. So we've got enzyme plus one substrate. And all of these things, the V naught, Vmax, Km, and the concentration of substrate, these are all things that can be measured experimentally. This slide will help you understand how all of these things come together and the different equations that we can use. So we already talked about the Km, that the Km is the concentration of substrate when V naught is equal to half the value of Vmax. You can see this tangent line. I'm going to highlight it. That's the V naught. The slope of that is Vmax times the substrate concentration over Km. Again, you don't need to memorize that. Just giving you a lay of the land. This asymptote here is when V naught is equal to Vmax. So when you're looking at one of these curves, you'll be able to kind of get your bearings. The x-axis is always going to be substrate concentration. So this is millimolar, but it can be any concentration units. So nanomolar, millimolar, whatever makes sense for your enzyme. And then we've got V naught. That is in the units of concentration per time. So in our example, we've got micromolar per minute. Whatever, again, makes sense for your enzyme. This is showing the derivation of Km, showing what it means. So when V naught is at half the value of V max, you can substitute in V max over 2 for V naught. This is just the Michaelis-Menten equation. So you're substituting in V max over 2 for V naught. When you do that and you simplify, it shows that the Km is equal to the substrate concentration when you're at half of your Vmax. Now what can we do with the Michaelis-Menten equation? So we can do our nice little plot after we've done a bunch of different substrate concentrations, keeping the enzyme constant. So same concentration of enzyme, different amounts of substrate. We can do all kinds of things to plot this data and look at it and get an understanding of how the enzyme works, get some values that we can do something with. So you can actually use some algebra to transform the Michaelis-Menten equation to a linear form. So this is the hyperbolic form. The other half of the curve is underneath the x-axis, so it actually looks kind of like that. But we don't actually concern ourselves with this part. Okay. You can actually just do the inverse of the Michaelis-Menten equation to get a linear form. I'm not going to walk through the math on what that looks like, but here it is. You can simplify that inverted equation, and then you get the Line-Weaver-Burke equation. You'll also see it called double reciprocal plot. Those are both the same thing. So you can actually graph this and you'll get a line. If an enzyme actually obeys Michaelis-Menten relationships, if you plot 1 over the V naught versus 1 over the substrate, you get a straight line that has that follows that equation that we just derived. And this slide is showing you the different points that are important here. So I'm going to circle them as I talk about them. 1 over the Vmax, that is your y-intercept. And if you don't remember your algebra with lines, you might want to brush up on that because you're definitely going to need it here. The x-intercept is the 1 over km, the negative 1 over km, okay? And the slope of this line is km over Vmax. So those are the 3 things that you'll need to remember: the y-intercept, the x-intercept, and the slope. All of these things will be helpful when we're doing some further analysis in part 2 of this lecture. Now, having Km and Vmax are great, but we need to be able to interpret them and kind of compare apples to apples. Enzymes have a wide variety of characteristics, and we need to be able to compare them. So Km can vary for different substrates, even within the same enzyme. So hexokinase, it can use ATP, glucose, fructose, but the Kms for all of these are different. So it's kind of difficult to compare Km because Km can mean something different depending on the actual mechanism of the enzyme. Now let's say that you actually have a reaction with 2 steps. The Km is kind of a catch-all for a more complicated equation that involves all of the rate constants. Remember, the lowercase k that is italicized is a rate constant. In this 2-step mechanism that we're talking about, we've got enzyme plus substrate reversibly forming ES, and from there we're forming E plus P. Now sometimes you'll see the this as just a single arrow where it's irreversible to go from ES to EP. But this is the 2-step mechanism that we're talking about. And to fill in our K values, this is K1, K minus 1, K2, K minus 2, which we're not really talking about here. Why does this matter? Well, the Km is relating the breakdown of the ES complex. So the ES complex has 2 choices. It can either break down and make the enzyme plus the substrate, so it just falls apart, or it pushes forward and does some chemistry and releases a product. The Km houses all of the rate constants that have to do with that decision of what the fate of the ES complex. So it's K2 plus K minus 1. So that's the 2 different routes that the enzyme substrate complex can take, divided by the formation of ES. If K2 is rate limiting, then that means that K2 is small, right? It's going to be smaller than the other rate constants. And if it's much smaller than K minus 1, then that means that we can essentially ignore it because K2 is so small that it's practically like adding 0. So in this special case, where K2 is much, much smaller than K minus 1, we can get rid of it and simplify the Km equation to K minus 1 over K1, which is defined as the dissociation constant, Kd, of that enzyme substrate complex. But this is a special condition. Cannot stress that enough. This is a special condition. It is not always the case. But under these special conditions, the Km represents a measure of the affinity of the substrate for the enzyme. But only under these conditions. If someone says that the Km measures affinity, blanket statement, always and forever, no. You tell them no, you shake your finger at them. That's wrong. Km is not always a measure of affinity. Go back to school. So we talked about Km and how to interpret that. Now we've got to talk about interpreting Vmax. So Vmax tells you the number of reaction steps and the identity of the rate limiting step, right? So all those things are going to vary from enzyme to enzyme. But Vmax tells you the velocity of your slowest step. And for that same 2-step Michaelis-Menten mechanism, The Vmax is going to be equal to K2, remember that's our slow step, times the total enzyme concentration. Because the total enzyme concentration, if you're under Vmax conditions, means that all of your enzyme is engaged and active. When product is released, it's rate limiting. And that is a little bit different from the Michaelis-Menten, going from ES straight to E plus P. It introduces this step of product release. So we now have this K3. And in a lot of enzymes, product release is actually rate limiting, not product formation. So everything up to here is very fast. And then everything after that is slow. So when we're talking about Vmax, it's not always going to be the formation of product. Oftentimes it's actually going to be the release of product, because that is the slowest step and that is usually rate limiting. But again, it depends on the enzyme. While it is common and it is a fair assumption to make, you cannot just go with that assumption as a blanket statement. It's like stereotypes. They may describe a certain portion of the population, but it is not true for everyone. It just may be common enough to where it becomes kind of common knowledge, but it's something that you should not expect to always be the case. Okay. So how do we look at KM and Vmax? How do we look at these things as apples to apples and have a fair comparison? Well, let's introduce Kcat, okay? It's a general rate constant that describes the limiting rate of any enzyme catalyzed reaction at saturation. Okay. So that means that the limiting step could be any step within this whole process, and we can call it Kcat. That's the understanding. So if one step in a multi-step reaction is clearly the limiting step, then Kcat is going to be the rate constant for that step. But things are usually more complex than that. And you can actually have several rate limiting steps where you have a lot of fast steps and a lot of slow steps, and all the slow steps are about the same rate. If we know that Kcat is the rate under saturation conditions, well, we just said that Vmax is equal to our K, it was K2 before, times the enzyme total concentration. But if this is the rate limiting step, then we can actually call that Kcat. So our new equation, let me do a color other than black. Vmax is equal to Kcat times the enzyme total. And if you rearrange those, then you get Vmax over the total enzyme concentration is equal to Kcat. Okay. And if you know that, then you can substitute in. And that is another very useful form of Michaelis-Menten when it comes to graphing your data. Kcat is also called the turnover number. So that tells you the number of substrate molecules that you convert to product in a given unit of time. Usually it's per second, but it could be per minute, if it's a slower enzyme. Catalase is something that usually you play with in biology or like a low-level chemistry class. It takes hydrogen peroxide and it makes water and oxygen. It is stupid fast, okay? Kcat, out of this world. Carbonic anhydrase is something you've also seen. That's in relation to your blood. So it makes bicarbonate. You don't have to know any of these kcat or turnover numbers. It just gives you kind of an idea of the range that you can see. If you compare the ratio of kcat over km for two reactions, that's the best way to compare, the catalytic efficiency. And that's usually what you'll see it termed as. You'll see catalytic efficiency, and they're referring to kcat over km. And what this does is it allows you to compare the conversion of enzyme plus substrate to enzyme plus product. If you have a substrate concentration that's much less than your Km, then you can actually substitute in and write kcat over Km times your enzyme total times your substrate concentration, and that can also be very useful for representing your data. So you don't memorize this. This is a combination of things that are helpful in the lab and things that you'll need to know for your exam. For your exam and just for your general knowledge, it's going to be much more basic, but I'm going into further detail here just because it's so rich. And if you're actually interested in doing enzymology, maybe you want to do an MD PhD program or you want to do research as a job, then hopefully this will give you some tools that you can be competitive in those markets. So kcat over km is actually a second order rate constant. So it's got the units of per molar per second. And the upper limit that you'll see for kcat over km is about 10 to the 8th, 10 to the 9th. That corresponds to the diffusion limit. So how fast something, a molecule can kind of wiggle around and travel and end up somewhere else. Okay, that's the diffusion limit. Kcat over km is a value that you can achieve with a mixture of kcat and km values. So you may have, a relatively fast, slow step, but, your km is a little bit on the large side. So there's lots of different combinations of kcat and km that can get you the same catalytic efficiency. Lots of enzymes actually catalyze reactions with two or more substrates. So Michaelis-Menten is great for one enzyme, one substrate. But large majority of them have at least two substrates and two products. So when we were talking about that covalent bond forming with the enzyme, we're talking about kind of two different substrates. You're forming a bond between the enzyme and some other group, like a phosphate group, perhaps, and then you can transfer that onto a substrate. So that would be an example of a covalent mechanism, and you have two substrates, the thing that's providing the phosphate and the thing that's getting the phosphate. You can also have redox reactions. You've got something being oxidized, something being reduced. So multiple substrates. And some reactions involve ternary complexes. So you've got enzyme, it binds substrate, it binds another substrate, and then it does some chemistry. So the Michaelis-Menten it's very powerful, but there are some limitations. You can kind of chop up an enzyme mechanism into pieces and make it fit the Michaelis-Menten model or theory so that you can do the traditional steady state kinetics. But keep in mind that biology is never that simple or easy. It's complex because we're complex. So you can have different modes of binding, like ordered versus random, and you're not responsible for knowing that. But just understand that there are mechanisms out there where enzymes do a lot of things that don't fit neatly into the Michaelis-Menten theory. Some enzymes don't even form a ternary complex. So you might have binding of a substrate, and then you make a product, there's some kind of change to the enzyme and that product leaves, then you have a second substrate bind, and then maybe your enzyme transfers a group onto that second product, or second substrate, excuse me, and then it's regenerated. So there's lots of different caveats. And again, not going to hold you to knowing all the different mechanisms like ping pong or double displacement or anything like that. Just have a general understanding that there are enzyme reactions that don't fall neatly into the Michaelis-Menten bucket. That's it for part 1. Make sure that you tune in for part 2, where we start with pre-steady state kinetics and continue on to talk about that, and we'll also talk about some example mechanisms. Be safe, and I'll see you soon.